All right, this is chapter 15, Senses, of trying to just recording a new format. I have in the basement. It's like a deep, deep cave, so I'm not too bright here. This is one of my favorite chapters to do because I studied a lot of biology in my life. I'm an ex-cancer researcher, and I never got to do, or I sometimes forget the basics, like how our senses work. And it came to me. I thought about it. I got a friend who's colorblind. He can't see the difference between red and green. And so I asked my friend Eric, I was like, what do you see? What colors do you see? He's like, I see all browns and grays. I was like, you can't distinguish red from green, but you know what browns and grays are. So it kind of confused me. I still think about that. So I guess we can go ahead and get started. All right. So this is what you're supposed to get out of this chapter. Of we have four different categories of sensory receptors, and you got to know the stimuli. Of you need to know the difference between perception and sensation, and of there is a big purpose on how our brain works on signal integration and sensory adaptation, and we'll go into that more later. All right, so we have sensory receptors. These convert different signals. They can be chemical signals, physical signals, of electronic signals from the environment called stimuli into nerve impulses. This conversion is called sensory transduction. We have exteroceptors that can detect signals from outside the body, taste, smell, vision, hearing, etc., etc. All right, and those are constantly being fed to your CNS or central nervous system. The list of the exteroceptors, you got taste cells, olfactory cells, that's your smell. You got these rod cells and cone cells in your retina. Hope you've heard that before. For vision, we got hair cells in our ear that help us hear. Uh, and then we have other small organs and cells basically in our ears that give us equilibrium or balance. Difficulties moving to my next slide here. All right, there we go. And then we got interoceptors. Those are signals from inside the body. Like, you can feel changes in your blood pressure or your heart pumping, things like that, your temperature. And so, we break down these sensory receptors and kind of the ligands or the signals they receive. So we have chemoreceptors, they recognize chemicals, photoreceptors, photons of light, mechanoreceptors, so that physical feeling or pushing, and then we have thermoreceptors that basically respond to temperature changes. All right, kind of what I went over before. All right, just the different types of sensory receptors. All right, so how sensation occurs, how do you receive that signal? Sensation is the conscious perception of stimuli, so it occurs in our cerebral cortex. All right, and so it gets signaled out from there. All right, just a little diagram of the central nervous system and peripheral nervous system and how they work together. All right, so we get integration or the kind of the summary of all these stimuli, and we adapt to stimuli over time, sensory adaptation. Um, smelling odor for the first time. The way I think of sensory adaptation is I used to live in Corpus Christi, South Texas. My community college students there, it'd be 100 degrees out there in black leather jackets and full black pants and boots. I'm sweating like a pig here in shorts and a t-shirt. Like, oh, go, Dr. Greg, you just get used to it. I was like, okay. All right, so if you want to check your progress here, you can. I would recommend just moving through. You can, unless you've done this slideshow a couple times, because you're not going to remember it all for the first time. Biology is a lot of review. All right, more learning outcomes. You can review those on your own. All right, so somatic senses. These are senses associated with your skin, muscles, joints, and viscera. And viscera to me is just your the outside of you. All right, and you got three types of receptors here. You got uh, proprioceptors, of cutaneous receptors, and pain receptors. The proprioceptors respond to mechanical stimuli, so they help maintain muscle tone, equilibrium, and your posture. They're associated with muscle spindles, Golgi tendon organs act together, give you muscle tone. They're responsible for the knee jerk reflex. So when you went to the doctor when you're a child, or even now, they take that little rubber hammer and hit you kind of below the knee. And if these receptors are working properly, your knee jerks. So there's this little diagram on how the different uh, mechanics work together, all the different cells and tissue types work together as well of these proprioceptors. All right, so we also have cutaneous receptors. Cutaneous means referring to the skin. So these receptors can tell us, if you've been touched, is there pain, is there temperature changes. So cutaneous receptors can be sensitive to different types of pressure, and these are due to certain structures in our skin. So we have Pacinian corpuscles, they're onion-shaped and they hang out in the dermis. We have the Ruffini endings, they're encapsulated by connective tissue. These are as part of the cutaneous receptor structures. And these thermoreceptors are free nerving ends in the epidermis. All right, so we also have pain receptors. All right, so if we're physically damaged, either by mechanical or chemical, thermal, you've electrocuted, we have pain receptors to detect this. All right, so there's different types of pain receptors. We have these nociceptors in internal organs, pain and skin as well. I'm going to pause this. I'm going to pass on this. Check your progress. Let's see. So, moving into taste and smell, there are certain structures and cell types you're going to have to be familiar with. All right. So, taste and smell are chemical senses. So we detect these little molecules in the air, and we on our tongue when we taste them. All right. Uh, so just structures, little small molecules that bind, have different smells. All right, so these chemoreceptors, your text divides them into two types. There's one more type, I'm not going to talk about that. So we have olfactory cells that respond to distant stimuli, so that's your sense of smell. All right. And then you have ones in your tongue, your taste cells, to direct stimuli. So you have to physically have the substance on your tongue. And then your tongue can tell you the pH, if it's spicy, if it's sweet, uh, basically separating out the different macromolecules. All right. So you have taste buds. They're on your hard palate, on your pharynx, epiglottis. All right. And humans have 5 main taste receptors: sweet, sour, salty, bitter, and savory, or umami, kind of like a meat flavor. There's the structure of your tongue and all the substructures to show you where your taste buds are at. And then on the right, that's that onion type cell structure we talked about before. You see pictures of them. All right. So how do you get the taste in your tongue to your brain to say it's sweet, salty, spicy? Taste buds have a pore. So pore is just an opening or a hole. All right. And they have taste cells as well. So it's not just one cell. There's multiple cells that work together, and they transmit the signal of basically to the brain. I guess in specifics of the taste cortex, and then you basically your brain analyzes what it is. All right, sense of smell, olfactory cells. Book wants to remind us that when we taste things, the taste is really 90% of signal still from the smell because it's so close to your nose. And so we basically breathe things in, and in the nasal cavity, it has these cilia which respond and give us our sense of smell. There's a diagram of it here. All right. So basically, like how good can we smell is what the text is trying to describe here. So it says each olfactory cell can only recognize 1,000 different types of receptor proteins or smells. All right. So in summary, an odor contains many different odor molecules, which activate a combination of receptor proteins. So basically, every smell has different numbers, and it's coded kind of like a computer. I think this is interesting that the number of olfactory cells declines with age. So I guess as you get older, you can't smell as well, which I think is true because I'm old. And it's also true that I think people, if they think about it, they recognize it, is the sense of smell can also trigger other sections of your brain besides what it smells like. So it can trigger different emotions from that smell or a memory. You check your progress there. All right. So now we're going to talk about how we see. All right. So your learning outcomes here basically structure of the eye, how the eye focuses on near and far objects, the different types of photoreceptors that we use in our vision, and maybe we'll talk about some of vision problems like red-green color blindness, which I discussed before. All right, so the eye has three layers: the sclera, the choroid, and the retina. The sclera is outer white, fibrous. Except for the cornea, it's clear. If you wash your hands really well, make sure there's no germs on it. You can actually touch your sclera or the white of your eye, and it doesn't hurt. So you can kind of leave it there for a couple seconds, press on it. You'll feel physical pressure, but it won't make your eye water or a sudden reaction to blink. But if you touch your cornea, which is the color part with your fingertip, it will hurt really badly. You'll start tearing. Your body will jerk back, and basically it's trying to protect that cornea because that's kind of where the lens is and all the good stuff. If you get a chunk of your sclera taken out of the eye by accident, like you got poked by a stick, you could see perfectly fine. It would eventually heal. But if you got poked by a stick right in your cornea of the wrong way, you could be permanently blind in that eye. All right, just a diagram of all the different structures of the human eye. You all can review that on your own. All right, anatomy and physiology of the eye. The lens is attached to what's called a ciliary body by these little ligaments, and it basically divides the eye into two different compartments. The anterior is the front, the posterior is the back. All right, and there's a lot of water in your eye. It's known as aqueous humor. All right, so something about glaucoma. It's basically, you get cloudy eyes. You can't see usually when you're older. Though some people who have them when younger and they have to get them surgically removed. But it's basically a buildup of that eye fluid, aqueous humor, and the fluid has substances in it. So when it gets, in front of your cornea or your lenses, it's kind of like looking through a cloud. All right, the retina is your third layer of eye. It's in the posterior of the back. All right. It's an area filled with the eye juice too, the vitreous humor. All right, holds it in place. Contains two types of photoreceptors, the rods and the cones. I hope you might remember that before. The rods, very sensitive to light, but don't detect color. So great for your night vision. Or you're going to see black and white with those. The cones require way more light, but they do detect color. So when it's very dark, there's not a lot of colors in your room, but with the lights you can see more. All right, so more substructures of the eye in the retina. So they have fovea centralis, is where all the cone cells are. All right, so when we look directly at something, it goes through the cone cells. We also have the optic nerve. It's just a bunch of sensory fibers from the retina. It's like a big cable that goes from the back of your eye basically up to your brain. And so if that optic nerve gets pinched or damaged, you can go blind. All right, here's just a table of the different structures in the eye. So you have the structures on the left, the function on the right. This is actually a really good piece of information because it actually breaks it all down. So in your slide deck, I would definitely print this out. That way you have it here, you have it as a reference. All right, so different functions of these substructures of the eye. All right, so we got the cornea, lens, and that aqueous juice focuses on the retina. All right, then light rays are kind of bent when they're brought into focus, and then you got an image on the retina, which is like the screen. However, it is inverted and reversed from left to right, and your brain processes that and then flips it around for us. All right, so this just basically says when we go from close vision when we're reading to far away, that we have to kind of change the shape of our eye through different muscles and different tendons. And if your eye changes shapes due to a variety of reasons and it's not the way it's supposed to be, then you don't see properly, and then you have to wear glasses like I do. All right, so just remember there's this thing called eye strain. All right, so you really shouldn't be on screens more than probably 4 hours a day if you can, or take breaks. It definitely causes eye strain. Just a diagram how light focuses on the retina and how the image is kind of inverted and not correct in the left and the right. All right, this is detail. I don't think it's that important. All right. So lots of details. If you want to talk about pigment or where the colors are in the eye and where synaptic nerves go into, that's fine. But just remember the eye has a lot of different cell types, but one of the most important is the nerves that run through it, and that optic nerve especially, because if that ever gets crushed or pushed on for a variety of reasons, you can go blind. All right, just some nice photographs of photoreceptors and the different parts of them. All right. So rhodopsin is a protein. It's a visual pigment in rods. All right. So it gives your eye color. And basically, remember the rods are very sensitive to light and are suited for night vision, but no color. The cones let you see the color. That's how I kind of remember it. Cones starts with a C, color starts with a C. So the cones, those photoreceptors, give us color, and the colors that these three cones can see are blue, green, and red. All right, so and all the combinations of that give the colors we can see. And obviously, this is actually, your text doesn't say this, but it's really breaking down different wavelengths of light. And so, we can see the blue, green, and red wavelengths with our eyes. There's certain wavelengths of light we can't see, so insects see a world very different than us, and certain lizards, depending on the wavelengths of light that your eyes are designed to detect and be able to process, and that's different in a lot of living organisms. All right, the retina, 3 layers of cells basically. We've got the rods and the cones, and the ganglion cells, which is your nerves that make all those connections. How all those are lined up together. So what this is, it's the structure of the retina. So on the left is a cartoon diagram. It's a 3-dimensional chunk cut out of someone's eye. And you can see the different layers and the different cell types. So that's just an artist's rendition of this. If you go to the image on the right, it looks like a microscope slide, so I kind of like that. That's not how it would actually be, but that's a histological section of, through an eye. I don't know if it's human or not, some type of animal eye. And what they do is they fix the tissue and they slice it onion paper thin, and it's just a single layer of cells. And they stain it with different dyes so they can basically demonstrate the different cell types on the slides. So that's called a histological image. All right, so we have a blind spot in our eyes. I don't think we can ever detect it or notice it, but it's basically right where the optic nerve exits the eye because they can't get any signal there. Just the structure of the retina. All right, so these nerve impulses that are carried by the optic nerves from the eyes. Then they go to their next step is the optic chiasma. Chiasma basically means X, so it's an X shape. All right, when the fibers cross. And then these nerve signals go to the visual cortex in the occipital lobe. Just a diagram of the pathway of how these signals flow from the eye to the brain. All right, so just a reminder, we have 2 eyes to give us depth perception, of I wear glasses and sometimes when I was younger I wore contacts more often, and I'd be in the middle of the day driving around and all of a sudden I'd lose a contact and all of a sudden my depth perception was off. You can survive, but you have to drive really slow and it's not fun. All right, so different abnormalities of the eye. So we talk about color blindness, very, very common in Caucasian population. Roughly 1 in 10 white males is red, green colorblind. All right, so another problem, which is what I first started with when I was in 3rd grade, I was diagnosed as nearsighted. So I could see things close up, but I couldn't see things far away. And if you're farsighted, it's just a flip of that. All right. Now I'm both. I'm basically blind, so it's like trifocals. I need readers. I need long distance. I mean, this is about as good as it's going to get. So, to that note, if you got a grade issue, sometimes I'm working on a little computer, and if I hit the wrong button on Canvas, it might change your grade. Don't freak out, okay? I'm not trying to kill you guys, so just shoot me an email. I'll look into it. And 90% of the time, I say the student is right. So there's a glitch, and we can make. And as soon as I get the email, I can make the change in like 5 or 10 minutes. So don't lose sleep over it, okay? All right. Just trying to show you how corrective lenses work. When I look through these glasses, how it basically changes of the kind of the curvature or the angles of how the light's bent. So people are farsighted. They can see far away, but they can't see close. So those people usually just buy readers. A lot of people like that don't even have to get a prescription from an optometrist or an optician. All right. Once again, how the light and image is bent. Some people in their distance vision might have what's called astigmatism. where there's a fuzzy image due to a variety of things, not just the corner of the lens being uneven. But sometimes this can be corrected through lenses, sometimes it can't. All right, just the bending of the light again, how it moves differently through all these different eye disorders. All right. All right, next is we're going to talk about the sense of hearing. All right. So, our ears have two functions. We always know about the hearing part, but it's also our balance or equilibrium. So a lot of times if you have an earache, you might be dizzy. The ear, the A&P of the ear, anatomy, physiology, it has divided in three parts of the ear: the outer, the middle, the inner. Generally, the outer is the toughest. The middle can take some manipulation, but the inner ear, you really don't want to put anything down there, right? Because if you bust your eardrum, it's not good, and sometimes it can be permanently damaged. All right, so the middle ear, the outer ear is just kind of the skin, and then the middle ear begins of where the what they call the tympanic membrane, or it's the end of that bony wall. All right, it's kind of hard to describe. That's the middle ear, and it has different bones there. All right, there's a diagram of your ear. All right, so you have a tube in your ear, basically to the middle ear of up to your nose. You got one going right out to your ear. There's different tubes, and the auditory tube is the one where people get tubes in, so that the inner ear doesn't fill with fluid. All right, sound waves come through vibrations. Basically, vibrates a membrane like a drum. Helps us hear things. So, in the cochlea, which is part of the auditory cortex, it has three canals. These different canals let different things pass. They have different hair cells and membranes, and then they have nerves that surround them all. All right, I'm not going to really go over how the spiral organ translates sound waves into nerve signals, but it's because I can't explain it. I can kind of see it. It's when the sound comes through, these little yellow things kind of vibrate, and that produces a chemical signal. So you can take that physical motion to chemicals and then shoot it through all the cells as a basically a nerve signal. All right. Also remember, the cochlea also can help us detect different wave frequencies or pitch. So if it's high or low sounds. All right, and also controls the volume. All right, so different things, you can review this on your own, affects your hearing. So basically when things are over 100 decibels, it's not good for you. And if you have this, throughout your lifetime, it will cause hearing loss. All right, so the other part of the ear is the sense of equilibrium. All right, so you need a sense of equilibrium so you're not always dizzy or sick. When it's out of whack, you'll have vertigo, which is when you're dizzy all the time. You might fall from it. This is really bad in older people because of bone breakage. All right, so to have a sense of equilibrium, we have a nerve that basically originates in canals in our ears, and they're connected, and it takes that signal right to the brain. All right, and these are basically mechanical receptors. All right. So we have this thing called rotational equilibrium. I think it's a big fancy word for when your rotational equilibrium is out of whack. It's when you've been on a roller coaster or one of those rides that spin and spin and spin and spin. And I rode the Catwoman whip at Six Flags, and it's the roller coaster that goes all the way around, and you're spinning and going backwards and forwards. So I felt very sick. So that disrupted my rotational equilibrium. All right. So once again, it's the way to describe, this these canals are. It reminds me of when I see, because I've dissected animals and ears, and we've looked at them underneath the microscope or a dissecting scope, and they look like little shells, and there's fluid in them. And if that fluid gets all mixed up and is on one side of the shell versus the other, then you feel dizzy and sick, and your rotational equilibrium is out of whack. And yep, as soon as that shell's all got is out of whack, it just sends signals to your brain saying, "Oh, you feel ill. Oh, you need to sit down. Oh, you're gonna puke or get sick." All right, just the mechanics on how that works. Of these sacs with these canals, they're located there. They also have hair on them, and they have these stereocilia, which are little tiny hairs. And besides all that fluid being out of whack, if those hairs get kind of triggered in the wrong way too, then that also causes us to feel sick. All right, so very similar rotational, gravitational equilibrium. All right, of when you're moved and things aren't kind of the right way, like when you come to a sudden stop in a car, you get another feeling, and this is caused by gravitational equilibrium. All right, so there's a group of cells called the stereocilia, and they basically bend toward the largest, the stereocilium, and they form this one big nerve, and that's how all these signals are transmitted. No way. We couldn't be done. Do we do all the senses? Oh, I need to think about that offline. All right, thanks guys.